Hello, everybody. Welcome back. I'm glad to present Dr. Ayelet Landau. Ayelet is an assistant professor of cognitive science and psychology in the Hebrew University of Jerusalem. Her research topics include visual cognition, the cognitive neuroscience of attention and time perception, and brain rhythms. So please welcome.
Thank you, Jonathan, for the kind introduction, and thank you, Neil and Orly, for the invitation to participate in this workshop. This is kind of quite a different crowd than the crowd I'm used to talking to, but I see it as a great opportunity to tell you a little bit about the type of work we do and the type of considerations my findings may have for the questions that you are dealing with at a somewhat different level. So I'm a cognitive neuroscientist. That would be my disciplinary affiliation. And as a cognitive neuroscientist, I'm interested in a basic question of how cognition, the behaviors, the experiences, our perceptions are embedded in physiology. Models, I mean physiological models, and I'll tell you a little bit about the models that I like working with. And specifically, as I will unfold later, my research topic is attention and perception, all concepts that I will define. Generally speaking, I hope that my talk is free of overuse of jargon, but since this is a different type of audience, I welcome you to signal if I'm using terminology that might not be familiar, because that's obviously an important way for me to communicate my findings. So as a cognitive neuroscientist, when I approach this organ, the brain, and I try to understand how the physiology is supporting behaviors, perception, and attention, to me, the ultimate goal of a cognitive neuroscientist is a question of integration. So it's clear that we have a lot of levels of description in the system and a lot of different scales, both spatial and temporal, that we need to overcome in order to orchestrate cognition or cognitive functions. And one framework, theoretical framework, that I find useful to work with has to do with brain rhythms. So generally speaking, when we measure population of neurons, it turns out that we can measure an oscillatory response from these type of populations. And by the end of the talk, I'm going to show you a few examples how this temporal structure can form an interesting consideration for when we think about what shapes our representations. And then maybe just one kind of more concrete note about this idea of brain rhythms. When I say is fluctuating in a certain temporal structure, I'm talking about electricity, so the field potentials are fluctuating, and neurons, which at the individual units level, become more likely or less likely to do that thing that we know that they're doing, so outputting action potentials or just the output that we often in physiology say that is what this neuron prefers, or even we might say this neuron represents. So when I say that we have temporal structure in a population of neurons, what you need to think about is the fact that we actually can kind of predict in time when content will be communicated or when content will be outputted in a given region. So just some historical notes about brain rhythms. They've been with us for a while. Already in the 20s, Hans Berger, a physiologist, actually a physician, has already documented the existence of brain rhythms, and these were brain rhythms that he was able to collect by putting an EEG while they were sitting idle or while they were sitting relaxing. And one thing that struck Hans Berger is that there seemed to be some structure. So this was the first evidence for temporal structure in physiological signals, and therefore it was coined alpha because it was the first. So if you ever wondered why bands and frequencies are named that way, alpha was the first to be discovered. It is also a very big and large signal, and you can even appreciate for yourself the periodicity of this signal. Ever since Hans' observation, there have been various levels of description that have yielded these type of measurements. So, when we measure population of neurons in animal studies, In humans, sometimes we have the opportunity to record responses from the surface of the cortex, and non-invasively when we use EEG or MEG, so magnetoencephalography. So these are all methods that allow us high temporal resolution in the measurement, and the signals are robust and ubiquitous in the sense that we see them in all these different levels of proximity to the substrate that we are measuring. And as the field has evolved in the past almost century now, many different rhythms of these periodic responses have been identified. So this is just a schematic from a paper from Georgi Buzaki, a very prominent researcher in this field. And I highlighted this particular range, which are rhythmic activity in the brain that range from around 1 hertz to around 100 hertz, broadly defined. And we call them different names. We have the alpha band, as I discussed previously, that sometimes abuts another rhythm called the theta band, the beta, the gamma, and so on and so forth. The reason I highlight these rhythms is because it turns out that these rhythms seem to play a role in cognition. So they're going to be players in what I will present to you. I'm giving you one example of what I mean when I say that we have cognitive functions or that these responses can occur and we can measure them by showing you a very simple example. So here we have an observer that is looking at the onset of this particular stripy stimulus. And if I were to measure her perceptual response in visual areas, and of course this is idealized data, I will see a gamma rhythmic response, which means that there's going to be oscillatory activity in a range of frequencies around 50, 60 hertz. So sometimes we look at it this way, and so this would be the response compared to some kind of rest condition. So this is frequency, and this would be some kind of unit of amplitude. And we can also talk about it in the time domain. So this response will be sustained as long as the stimulus is there. We will associate this response with the stimulus because it's sustained and lasts as long as our stimulus lasts. And one small note for the maybe a few physiologists that are in the crowd, an important feature when I'm talking about an oscillatory phenomena or about brain rhythms is that I'm looking at something that is narrow band. So I'm not just looking to see this kind of profile that is really centered on a certain frequency and is sustained, as I mentioned. So when I look at this kind of ubiquitous finding of brain rhythms, as a cognitive neuroscientist interested in perception, one of the main questions that drive me is what is the cognitive relevance, or what is the role of brain rhythms in cognition? And I'm going to exemplify a few different type of roles that, that these signals may have by looking at work on attention and perception. I lump them together. And I think there's some utility to that. So think of attention as a case study, a various cognitive function that is our case study to look at what can rhythms do for the system and how can they constrain our perceptual representations. And I'm going to blow my thunder by telling you the 3 points that you're supposed to walk away with from this talk right away. At the first level, I'm going to show you attention are implemented through this rhythmic brain activity. So the same gamma response that I've shown you as an example for when do we see a rhythmic oscillatory response is actually serving as a communication protocol, if you will, for attention. And don't worry, I'll explain what I'm talking about by saying attention in a second. And I'm then going to move on and talk about two other interesting constraints that we have in the system, which is that perception, turns out, is also a rhythmic process. And finally, I'll talk about work that I've done looking at distributed attention and how it also utilizes a rhythmic temporal profile. So what is attention? Attention has a few ingredients, and the definition should be quite clear, and it's very nicely exemplified in almost any everyday example. Our environment is rich. It's way too rich for us to actually process it all in full. And what this means is that there's always things that are competing for our resources. Now, I could take you to the beach, and I could tell you that your friends are waiting for you under a striped parasol. And what your system has evolved to be able to do is to single out a location that is relevant and process it in a prioritized fashion. So that is what I mean when I say attention. So there are two components here. One is the environment produces these competitive interactions. The second is a selective process in which when something is relevant, you are able to basically prioritize one location, and that process also prioritize the rest. Now, what does that look like when we're talking about the physiology? Because that is really what we're interested in. And turns out there's quite a clear description from this everyday example to the level of a receptive field that I could provide you with. So, so these are, so now basically what I'm going to show you is these two-stage process. One is the competitive interactions, stating basically the limitation on our ability to process everything, followed by how attention can actually resolve the competitive interaction. So we're going to go into, we're going to kind of take a scaling, and we're going to scale into a receptive field of a V2 neuron. V2 is a visual area, one of quite area in the system. And when I say receptive field, I mean the part of the environment that a given unit is responding to. That is what I call a receptive field. So the receptive field in this plot here is going to be denoted by this dotted line, meaning this neuron responds to anything that is in this dotted line. And you see here that there are two different stimuli that we can present to the animal or even to this receptive field. We can present a vertical line and we can present a horizontal line. And they don't give us the same response. So we now know something about this receptive field in this particular region, which is that this particular neuron has a preference, something that decades of research does. People measure preferences of neurons. And in this particular case, this neuron prefers a vertical line. It does respond to a horizontal line, but less so. Now, I promised you some competitive interactions, so what we're going to do is we're going to present the same receptive field with two stimuli. And lo and behold, what we see is that the response to the two stimuli falls somewhere in between. Now, I didn't ask you the rhetoric question of how does the combined response have to look, which might have been a better path to take. But when we think about what a response to two stimuli should look like, we could have thought alternative models. And I think the more intuitive models would be either that the two responses would be added in a kind of additive way, combined in an additive way. Or you could have thought, look, this neuron prefers its vertical stimulus, then it should just give that response because there is a vertical stimulus in the screen. But that's not the case. There is some weighting of the two. And that is what competitive interactions look like, or that is what too many stimuli competing for resources looks like when I take you into a receptive field of a neuron. Now, this is a talk about attention, so how can attention help us out of the situation? Because if you think about it, when you're walking around in the world, this reality means you're not processing optimally, right? Because if we kind of took the project of the past decades of describing preferences of neurons, but then the world has more than one thing presented to these neurons, it means that you're all the time walking around in a suboptimal level of representation, okay? But then attention can come to help us. And this is basically what I'm laying out here is a theory called biased competition theory. And so now our animal will have attention directed to this one stimulus, and albeit both stimuli being present in the receptive field, the response or the preference of the neuron, if you will, has been recovered. So don't worry, if you're able to pay attention to things, it doesn't mean that you're walking around processing the world suboptimally. So this is what I'm describing as attention at the receptive field level. And of course, as many of you may know, the visual system has a hierarchy. So I could take this depiction and kind of lay it on a very schematic hierarchy as follows. And I told you to pay attention to the parasol. And my lower visual areas will have receptive fields that are going to be very small. It means that each of these stimuli has their own receptive field. So there's no competitive interaction there. Now, we do know that as we move up in the visual hierarchy, the receptive fields grow, and now we're in a competitive situation. I told you to look for your parasol. So I know that this higher visual area is now going to be representing the attended location only, and the attended stimulus only, and not the unattended stimulus. So that is exactly what I showed you in the previous slide, except now I'm not focusing on one particular region with a fixed receptive field, but I'm taking this whole principle and saying, look, even though this higher visual area could encompass both stimuli, what I just showed you means that when I'm paying attention to one, its representation is going to be limited to an attended stimulus. Now, at the outset of the talk, I told you that every response of a population of neurons to this kind of visual stimulus is associated with some rhythmic brain response. So, okay, I'm going ahead of myself. So the question is, how is this mechanism implemented in the brain? How has it come to be the reality that this higher visual area is only included in attended stimulus and that the unattended stimulus is curtailed? So as I said, we have an oscillation in each one of these regions. So this signal, the red signal, is associated with this response, the blue with this response, and then the higher visual area is producing the oscillation that is signified with this purple signal. And it turns out that, so we said there is a gamma band response, and I also indicated to you the fact that this close from their ability to output. And the outputs in this slide are signified with these lines. So these would be spikes, okay? And it turns out that when we pay attention to one location, the fact that it is propagated through the hierarchy is supported by the temporal relationships between the signals in the lower visual area and the higher visual area. So if you will, the reality is such that a lower visual area can drive the next region in the hierarchy effectively, and it does so through this rhythmic brain activity, and that is what promotes that representation on. It is also sneakingly the way in which this other representation is basically missing out on the window of opportunities to actually communicate something meaningful, its outputs, for further processing. So that is just the first example on how basically the neural mechanisms of attention are implemented through rhythmic brain activity. This temporal structure is used in order to create effective connectivity between different regions and therefore overcome the need for spatial integration, because we know that there is a hierarchy and we know that there is a certain propagation of processing that allows us to function on whatever our perceptions are. Another goal or interesting question is, now that kind of we're familiar with this structure, I would like to introduce a perceiver in between, right? So all that drama with the gamma band activity at these high frequencies occurs at the neural level. And what I want to touch upon, pioneered by the group from Ruffin van Roolen in Toulouse and others, that basically asks, okay, is there any temporal structure in perception itself? And I'm not going to go through great detail to show you the data. I'll just give you the bottom line that comes out of this kind of work, which seems to show a very clear picture that if we're just looking at this higher visual area, which I kind of associate with our ability to respond, or even this level of representation would be the level most closer to our awareness of the stimuli around us, that representation, it is fluctuating. So now I'm not talking about the fast fluctuations or the fast temporal structured gamma rhythms, but rather this is a slower fluctuation more around the alpha rhythm, so around 10 hertz. So you're looking at this parasol, you all feel that it's coming continuously at you, where in fact I just told you, and this is work you can read about from Magneticencephalography, told you that this isn't the case. It's not coming at you and being accumulated continuously, but rather it is fluctuating. And there are moments in perception that you're going to be better at detecting a faint target, and there are moments in perception that at which you're going to be worse. Now, the way they have shown this is they basically were able to associate an oscillatory brain response to a perceptual outcome. So they managed to show that an alpha rhythm in EEG measurements predicts the outcome, whether a subject will see or will not see a faint target. And of course, they're using EEG, but they're using EEG as a proxy, really, because what we need to be left with is the fact that perception itself is a rhythmic process. So other than reading these papers and going into the detail, I thought that sometimes when I show this to audiences, they get a bit resentful because this is nothing that we experience, right? I'm just telling you that your perception is going at 8 hertz. And obviously, our reality enables us not to experience that, right? So these processes have been identified with very, very faint targets and so on and so forth. And by and large, either properties, temporal properties of our reality, or potentially even interactions, cortical interactions between different units might suppress this sensation or this kind of experience. But there is a stimulus that helps bring out this, this experience, which I like showing because it has been actually alpha. So I'm going to show you now a demonstration that may elicit this kind of experience of flicker in you. So then maybe you'll feel a little less resentful for this idea that you're not getting your inputs continuously. So please follow and track with your eyes the blue dot as it revolves around this spokey wheel. And as you do that, just try to pay attention. Don't move your eyes from the blue dot, but try to pay attention to what happens to the center of the wheel. And of course, you know, the projection conditions are not ideal, but most of you should be experiencing some sort of flicker. Who's not experiencing the flickering of the middle of the wheel? I think most of you are, and experience your very to come to my laptop later. But the idea is there's something about this stimulus that, that breaks down the suppression. And there are other conditions in which we can actually demonstrate this and potentially even have people kind of experience their own alpha. So perception, we have to accept, is rhythmic. Now, in my own research, I'm interested in attention, and a simple question I wanted to ask is, does distributed attention also fluctuate rhythmically? Another motive, I actually wanted to directly be able to measure these type of fluctuations in behavior and not have to use the EEG as a proxy, right? Because theoretically speaking, if perception is a fluctuation, then I should be able to actually show that the benefits of being in a good moment versus being in a bad moment could be measured directly in asking people about their experience. So attention here, if you will, is an interest in of itself, but it's also a little bit of a trick, because when I'm talking about distributed attention, what I'm really talking about is introducing a competitive interaction, if you will. Right? So now people don't only pay attention in this schematic to the parasol, but they also have a Ferris wheel, right? You're in this situation, and of course you have a subjective experience that you're able to distribute attention over two locations. And what I want to know is, how do we resolve this kind of situation? And is this rhythmic characterization that I just demonstrated over kind of perception on a single location, does that govern the way we explore our environment or the way we distribute or negotiate this competitive interaction between two different locations? So I didn't take people to the beach for this experiment. I actually had them sit in front of a monitor and stimuli that are much like this, to pay attention and to detect a very faint target. So the target comes up, it's very brief and it's very faint. Critically, I don't have EEG signals here. I want to measure something directly in behavior. So I used an experimental trick to be able to anchor attention at a given time, at a given location. And the trick I used, I call it a reset event, but it's basically these 4 dots that are flashed very briefly in one of the locations. So now I'm going to just show you what the design looks kind of in one screen. So these are the gratings, we call them, these 2 patterned circles, and they're up for almost, for a very long period of time, so almost 4 seconds. The flash event is a critical event because only at that moment in time, decades, that when I flash something, then I clearly attract attention to this flash. So I know where attention is at that particular moment. Now that I know where attention is, I can start measuring performance at that location as a function of time to this reset event. So now I'm going to be, so these bars represent possible conditions in my experiment. At each trial, so this is kind of at each go, let's say, there's one target to be detected. It's unpredictable in time to my subjects, but by design, by the end of the experiment, I have all these time points as a function of this reset event. It's a very simple idea. I reset attention to one location, and now I'm measuring finally in time how people perform, okay, at that location. Now, I'm interested in attention, and a hallmark of attention is that it's spatially selective. I'm not interested in some kind of generalized arousal effect, and therefore, I don't only present my brief target here, I also present it at the opposite location, right? And there's a prediction to be made, right? If I am asking whether perceptual rhythms support the distribution of attention, what I'm hypothesizing is an alternation, right? Basically, attention should be kind of anti-correlated in these two locations. So this is exactly, so this kind of schematic is exactly how I present my data. So this is the reset moment, and what I'm showing here is accuracy. So how well do people perform? Do they have better and worse moments? And so these are the data I get. I'm going to ask you to ignore this whopping big masking effect. It turns out, and this would be a completely different talk about awareness, turns out that if you flash 4 dots around something that contains your target, you make people blind at that location. And that's not the phenomena that we're interested in. We're interested in seeing whether we have better and worse moments unfolding after that reset event. And it looks like we do. So it looks like performance at that location become better, then worse, then better, then worse, and so on and so forth. And that is at that same location. We could ask, of course, here too, we're seeing some sort of ebbing and flowing in performance. So on the face of it, it looks like attention is indeed fluctuating. I'm talking about fluctuations, and computationally we would prefer to describe these type of phenomena in the spectral domain, so to look at what rhythm does this fluctuation assume. So now I'm plotting here frequency and some arbitrary unit of amplitude, and what I found is that there is a 4 Hertz fluctuation in performance at each of these locations. Is performance correlated between the locations or anti-correlated is another question that we can probe our data for. And that is by looking at the phase relationship. So if the performance is correlated, that means we have a generalized fluctuation. It's probably not attention, maybe something like arousal. But if we are actually showing alternation, it means that this is a spatially selective phenomena that then we expect to see kind of an anti-phase relationship. So this is the average phase that we have. Right? These are time courses of performance. So this is people behaving. There's no physiology recorded here. So to summarize this part, what I was able to show is that, that we have rhythmic sampling, that when we distribute attention, we are actually alternating between different locations. We're doing that rhythmically at 4 Hertz, and that is consistent with the previous finding that perception is an 8 Hertz rhythm. Because if you think of it, now I'm taking my 8 Hertz sampler and I'm distributing it. And so at every given location, I might find a frequency of half that rhythm. Now, I don't know, in your field, maybe replication isn't such a concern, and mine kind of is. So every time that my findings are replicated, I kind of feel like it's my birthday. And I'm not going to go into great detail, but the idea of the use of resets in order to measure rhythms in performance and temporal properties of performance have been adopted by many groups all over the globe. And I don't think I have time to talk about all these replications, but it has been expanded in interesting ways, both in the field of attention, both in field of different types of attention, so object-based attention and other types of attention, and as well as, well, I really shouldn't go into great detail, but the point is there have been several replications, conceptual replications, demonstrating that indeed attention seems to entail this rhythmic sampling, replicating the finding schematized here. But at this point, I want to turn to another line of evidence that I have collected, which has to do with a critique to this finding. So I told you about the reset. It was kind of a smart trick, and it really enabled us to measure the temporal structure of performance. Okay, so maybe it's kind of also an important theoretical point. The assumption is that perception is oscillating all the time, and attention is basically the utilization of this sampling rhythm to whatever it is that is your goals at the moment, or that you're distributing attention over at the moment. And for me to be able to measure it, I need to use this reset, because it's a measurement thing. I don't know where I'm at in terms of the exploration of my subject at that point. But of course, this might not be the reality. It's quite possible that my reset event is kind of throwing a stone in an otherwise completely flat pond, and I kind of perturb the system, and now I'm showing this rhythmic dynamic, and it's not, in fact, this kind of ongoing rhythmic dynamic or temporal structure. So in order to address this question, I turned to physiology, and specifically to magnetoencephalography, which is a method similar to EEG. And I do away with a reset so that I'm not throwing stones into any ponds. And now I could actually get an ongoing assay of brain rhythms. And so in this part of the talk, I'm kind of going to try to tie together two different pieces of information that I've told you about. The first is, let's see if this 4 Hertz is an ongoing phenomenon. And then specifically, what I'm probing with my measurement is a signature of perception. So if you remember from the beginning of my talk, so now I'm in MEG and I'm measuring different visual fields, which we know are represented in the contralateral side. So I kind of have a map. I know where to look for which response in the brain, on the cortex. But specifically, I also know that there is another temporal structure in neural responses that I told you is the perceptual signature of a stimulus that is presented, and that is the gamma band activity, right? So we talked about the fast kind of gamma band activity that is a communication protocol in the first part of the talk, and now I told you that attention is a rhythm, it's a slow rhythm, and the question is, do these temporal structures interact somehow? So that's kind of one way to look at it. A more shallow way to look at it is, can we track the 4 Hertz attentional sampling in this signature? So then we would just use this as a signature. You could have given any signature. But I think for this audience, it's quite interesting that we're talking again about temporal properties and again about a potential of integration, now not between different regions, but in time. So this is kind of another potential. And that is exactly what we measured. Here, we don't have a reset event. So the way, the general approach for looking for this type of dynamic is by presenting targets in long trials, very similar to the ones that I've shown you. I don't have a reset now, so what I do instead is I actually look at the neural response that precedes successful trials and that precedes unsuccessful trials. And so now what I'm showing here is kind of my hypothesized data and possible type of trials that will go into my measurement, which is, okay, let's say I presented a target in the right visual field and I got it. So I can now look at what happened just before that hit in the contralateral hemisphere, and I could look at what happened in the ipsilateral hemisphere, so that's the other hemisphere that isn't representing, and I could try to kind of see what is the dynamic. So there is an extra step. This is not going to be overly technical, but the extra step is what I want to measure is some kind of temporal stressing of processing between the hemispheres. So in order to do that, I subtract, I take the difference between the representing hemisphere, if you will, and the non-representing hemisphere, okay? Because I want to see whether there is something just before that target that will predict whether my subject will perceive or not the target. If someone's interested in the analysis, we can obviously look into it later. And I'm hypothesizing that the 4 Hertz that I found in behavior will be that envelope that will govern my perceptual signals and predict whether a subject is able to detect or not able to detect the target. And what I'm showing you here is half a brain. It's half a brain because I subtracted one half from the other, so I'm left only with one hemisphere. So it's not that, it's not that we're hiding something in the other half. And what you're seeing here is a region that I have found that kind of fulfills my hypothesis. And the region that I found that fulfills my hypothesis is right around where the sources of these perceptual signatures are. And now I could ask, okay, so what is the phase difference between the 4 Hertz component that I found in this region? And basically, what this data shows is that if I look at this region, it is fulfilling this 4 Hertz fluctuation that I found in behavior or it governs and shapes the fast fluctuations that, that I've shown you are signatures of perception and protocols of communication between different regions. So, so last part about the brain here, one can ask, okay, you've shown us a blob, you kind of glossed over the analyses, but I'm kind of hoping that you walk away saying, okay, she found 4 hertz in behavior, and then she found that the perceptual signatures that are also rhythmic are kind of riding this 4 hertz. How much better are we when we're in the good moment of that modulation versus the bad moment? Because that's really what matters. If I just showed a little incremental difference in perceptibility, that's not so interesting. And so what I could do is I've defined previously and say, and basically kind of look at the phase of this 4 hertz and plot accuracy as a function of this slow fluctuation phase in my gamma. And what we find is something like a 16% modulation, which is in our field is considerable. So you're 16% better, or I can predict benefits in your performance that account for 16% effect size. So to summarize and wrap up both the last 2 sections, a rhythmic pattern, we see this in behavior. Visual information isn't continuously and evenly accumulated, and the periodicity is 4 hertz if I'm distributing attention over 2 different items. There is a seesaw effect. That's kind of a big deal for some portions of our field. We can't actually, in parallel, process 2 different locations. We actually do this kind of serial. We're better in one location, we're worse in the other, and vice versa. And attentional sampling is an ongoing phenomena. So my use of this kind of reset event likely reflects this reality rather than this reality. We did that by looking at ongoing signals without a reset and finding a similar dynamic, basically mimicking the behavior and retrieving it from this perceptual signatures. Another interesting aspect is that it turns out that we're not the only species that explores the environment in this kind of temporal structure. So if you look to non-primate as well as non-human as well as human, our eyes at around that kind of rhythm. So if you want, you could think of attention that I'm talking about sitting atop of some kind of hierarchy of exploration behaviors. Rodents sniff, whisk, lick at around that frequency. So there is kind of some linkage to different species that have similar functionality, and I personally find that an interesting link. So kind of there is consideration for temporal structure in arriving at many different representation types in many different species. How are we doing for time? How are we doing for time?
You got 6 more minutes.
Okay. Okay, so in that case, I think I'm going to walk one extra theoretical step, which has some implications for where this is going and for kind of the general consideration of temporal properties. And that is, now that we know that attention is a rhythmic process, can we go back to the receptive field? Like how deep does it go in terms of the scale of description of attention? I kind of simplified the reality and said, okay, this V2 dynamic, I showed you that is attention, and now I'm telling you attention is a rhythm, or competitive interactions are a rhythm. So I will go back to exactly this figure, specifically this figure, and propose kind of an idea that we're exploring these days. So basically when people do these measurements, often the measurement and the visualization and the analyses as well as the experimental design could be very much guided by the dimensions that the particular researchers consider. I hope that by this point in the talk, it's clear, well, to me it's obviously clear, but even to people who are doing kind of the more philosophical assay of these questions, that the temporal domain is very well worth considering, and there's quite an interesting dynamic that we need to think about. So I go back to these data which took no consideration for the temporal domain, and I would like to propose, oh, and I should say that this red line provided a lot of modelers a lot of work, right? So the idea of why do competitive interactions look exactly like that? People came up with very sophisticated models, and now I'm talking about computational models for how that comes about. But I would actually like to propose that after my talk, we might consider the following model, which is maybe there are temporal dynamics that this measurement in the design and in the analysis are not allowing us to see. So if you think if you let your mental calculator figure out what a reality that looks like this might result with, particularly if it's phase shifted, because you never know where attention starts, you kind of end up with this line. So it's quite possible that, that the models that exist for these competitive interactions, even within a receptive field, need to take more seriously the temporal considerations, try to look at finer grain temporal characterization of this type of dynamic. And what they will what they will learn is that what you are seeing here is a combined representation of a preferred and a non-preferred stimulus, except there is a temporal dynamic, and it is kind of bouncing back and forth between different responses. In my lab, so rhythmic sampling might actually qualify this physiological model of biased competition. In my lab, we're getting at these questions for now, mostly from a behavioral perspective. But if you think about this, then there's a lot of interesting predictions that could be made. One prediction is that we should be able to see rhythmic sampling even when things are overlapping in space. So I've taken kind of to the easy case of spatial attention, and I'm telling you there's this kind of seesaw. But if I'm claiming something about a single receptive field, then I need to deliver something a little bit stronger. I'm going to go very fast over this, but these are experiments that we've already been running, and we have these kind of objects. Someone mentioned moving dots, so they primed us. But these are moving dots that are defined by their color and by their direction. So if you want to think of it as two overlapping objects, and this is very fresh data, basically demonstrating that if we look as a function of a reset at performance, just as we shown it before, there seems to be a fluctuation. We could look at the spectrum and see that there seems to be temporal structure, even when we're kind of trying to process two objects in one single location, and lo and behold, we look at the phase relationships, and those two seem to be anti-correlated. So the temporal dynamics that I've demonstrated over space seem to also generalize over the case of what we call in the field feature attention, when two objects are overlapping in space but defined by different features, color, motion direction, and so on and so forth. And this is just one kind of the tip of the iceberg of a line of work that will be able to establish to what degree, for example, in the visual hierarchy or in different type of visual representations, do we actually see this temporal dynamic. And this, too, is a step along kind of a bigger research program that is trying to say something about these type of rhythms, providing a domain general mechanism for perception and attention. So along those lines, and I don't have time to show data for this, and some of these data are being collected as we speak, we're also looking at different modalities. I started off by saying that my job is to integrate different aspects in the human brain in understanding cognition. And my kind of one of the general projects in the lab is to basically inquire whether these temporal structures can provide a very powerful medium. I like the case of attention, because attention is that type of thing that really does need to harness information from various different sources. I considered the case for space separately. I considered the case for features separately. But you could also ask about how do we combine inputs from different senses. And so if you think of a kind of an exploration rhythm, qualifying, shaping our interaction with the environment, shaping, or if you will, shaping the state of the system as input is coming in, I think it's kind of an interesting idea to throw at you, crowd, who are interested in information and other considerations that, if you will, shape the representation and this interface between the external environment and the state of the brain. So I think this is a good time to stop. I want to thank you for your attention, to thank my collaborators in Frankfurt with whom I did some of the work. This is my lab. It's kind of brand new. The furniture is probably being installed as I speak. So until then, this is how our lab meetings look. But happy to take questions.
Okay, so we have time for question now. Oron?
Yeah, a more philosophical question. Don't worry. So you show that the temporal structure of attention and perception, that it has a rhythmic structure. And I just wondered whether you think that the fact that it is rhythmic has some sort of cognitive advantages rather than, let's say, the structure was linear, or it had other shapes. I mean, why being rhythmic is so special? I mean,
so it's easy to answer this question if you consider physiology, because in physiology it's quite clear. If you have a rhythmic temporal structure, you have a mechanism to interact between different regions, both promoting certain connections and curtailing others. And we know that these kind of, so that is one level. The other level is that, you can consider the integration between different temporal structures. So, I mean, the question of like, where does the sampler come from is kind of still under investigation. But if you need to, for example, even think up the situation that I depicted in the first part of the talk, where you have these two regions that are engaged in this kind of synchrony. You have to come up with a way to I'm going to say periodically allow opportunities to change the state of the system. And the more predictable this dynamic is, the more plausible it is that different systems could kind of get engaged and disengaged, right? So I think generally speaking, periodicity provides some kind of predictability in the system. So if you need to think of a system coming online and going offline, it would be nice for the system to be able to predict time windows at which a state can change.
Can you get the same sort of 4 Hertz anti-correlation between two stimuli or attention stimuli if you put them up and down instead of left and right? Do you know?
I haven't tried. Theoretically, I would say yes.
And if you can get that, wouldn't this predict that if you put 4 stimuli on 4 corners, it would go down to some function of 2 Hertz?
Correct. So that's exactly the prediction. I glossed over the replications, but one of the replications actually that utilizes a completely different method done in a completely different hemisphere on the globe has actually shown exactly that. So they had a very particular experimental setup that allowed them to check rhythmicity at 1 relevant object, 2 relevant objects, and 3 relevant objects. And what they show is that this more or less 8 Hertz sampler seems to divide exactly as you outline.
Wow.
Yeah.
Neil?
Thank you very much, Ayelet. It was very interesting. Of course, so you said, I think at the start that you showed us the example with the parasol being the target stimulus, and then you said something like, when the higher visual area has attended to that stimulus, we can see the representation at the lower areas, but not in the higher visual areas. So I'm wondering whether it's just in a particular section that's devoted to, I don't know, attentive processes, or in general you're saying that there are representations at the lower level but not at the higher levels.
The idea is that the visual system is a hierarchical system, and you have lower visual areas that have smaller granularity, so smaller receptive fields, and as you walk up, the receptive fields grow, right? So they become bigger, and you're basically looking at a larger portion of your environment and exposing the system to more competitive interactions. So we're not going to look at the lower visual areas because they're kind of behaving like a sensor of a digital camera, if you will, right? You're going to basically see in those lower visual areas anything and everything in the environment. But the crucial question or the crucial receptive field to look at is that receptive field that has converging inputs from 2 separate lower visual fields and into 1 upper visual field. And I keep it schematic, but that actually holds up for quite a number of levels in the hierarchy.
Rosa?
Thanks. So I was wondering why you needed the reset event. So it seems like if we have this 8 Hz sampling going on all the time, then it should be going on all the time.
It's as I say, it's a measurement trick, right? So if I want to measure it directly in behavior, if I want to measure the rhythm directly in behavior, I never know where attention is when I'm starting to measure it, right?
But why do you care where it is when you start? As long as you measure it for long enough, you should see.
I'm measuring accuracy in different trials. So that's kind of an important point. My time courses are an aggregation of many, many, many different trials. So if every trial, my sampler is at a different phase, and I aggregate many, many different trials, I will be canceling out any systematic fluctuation.
So if you just look in a single subject and you measure how well they
So then, assuming that single subject is consistent with where they start, you potentially could see that without the reset. I don't know that we can actually assume that a subject always kind of starts their sampling at a given
beginning of a trial.
Well, at a given location, right? I have 2 locations, and so that's kind of the issue. I have 2 locations. They may decide to start exploring on the right side or on the left side. So if all my subjects start exploring in the same location, then I wouldn't need that. Absolutely.
But in principle, you could do this with just one location, right? Just
Yeah,
one detection.
that's true. Correct.
And so, do you see a difference in?
I haven't done that experiment because in many ways, even though I mean, it's kind of every now and then we think about doing that experiment. The point has been made with the EEG studies because they have done the one location experiment. They haven't they haven't looked at behavioral time courses, but rather linked performance to the ongoing physiological response. But it's absolutely I mean, that exactly should be the case. Prediction.
So, yes.
Hi. Thanks for that. I just want to press you a bit on this claim that, you know, the brain rhythms is like the kind of key integrator of different levels here, because that seems quite sort of bold to me. So I mean, one way of pushing...
It is a mechanism. I'm not
one way of pushing back against that is to say, you know, look, it leaves a lot out. Right? And I can illustrate this by say, imagine we're trying to figure out how a computer works and integrate different levels. And I say, oh, when it does addition, you know, this signal of such and such a frequency is generated. When it does multiplication, we get this other frequency, so on and so forth. I mean, there's a whole lot left that we don't understand, right? About the way the system works and how the different levels are integrated. And you might say, it's not clear to me that we'd have to pass through a kind of intermediate stage where we get information about like the frequencies with which these, you know, tasks are carried out in real time in order to move from a kind of high-level description of the system to a mechanistic description that sort of tells us how that high-level capacity is implemented in lower-level units working in some organized fashion.
So are you asking, just to clarify what you're saying, are you talking about my kind of connection between the fast rhythms and the slow rhythms? Is that what you're
I see the data you're presenting. I mean, it's an interesting set of correlations and they're interesting signatures. It's less clear to me that it's just not another piece of data that needs to be integrated, perhaps via some other means, as opposed to the kind of key integrator, you know, that will allow us to
I'm not measuring a key integrator. So maybe this is actually a very important point. So the way I do cognitive neuroscience, and this is maybe an important disclaimer, is I like to work in systems that are very well understood and with signatures that are very well documented, right? So if I show you the narrowband kind of, I think I showed this at the beginning, oscillatory response, and now I'm talking about the kind of, here we go. So if I show you this right here, this part, this rhythmic activity, I can measure it. I could stick an electrode in a monkey and listen to a population of neurons. I could stick my subject into an MEG and measure this kind of narrowband activity. And this is what I would call a signature.
Yeah.
And so that is kind of a very basic level of description, which is, I think, hard to argue with, right? If you have that response, then you know you are driving that relevant substrate, right? And we know how to look at that. And then there's a second level, which could talk about, okay, can this the fact that this, because I'm showing something, I think, reasonably narrow band. So can this oscillatory response do something in the system? For that, I didn't bring in great detail, but we have the physiological studies showing that indeed you have increased synchrony. So you have this selective synchrony that, you know, in this scenario where you have the two receptive fields converging into a larger receptive field, you will see the stimulus that is represented in the attended receptive field showing greater synchrony with the higher receptive field, and nothing of that for that other input into the receptive field that is representing a non-attended location. So this is, and these are, you know, findings not from one lab, but from multiple labs. So these are things I feel quite comfortable with. Now, you point to like the key generator of this whole dynamic, and I'm probing the system from this kind of perceptual end. And I don't hypothesize, actually, that the generation of that happens there. So maybe we actually agree that what is the sampler, how general its scope is, those are the questions we're getting at. So, you know, I'm starting from the very low level of let's look at a perceptual signature. Let's look at how people's perception actually modulates. Let's try to see whether the two relate. Let's try to track my little 4 hertz in this type of signature. That is, that's an endpoint that I feel that my measures are not just another data point that you have to kind of integrate coming from Mars, right? These are kind of very solid signatures. The question of the generator, I think almost purposefully, I'm actually probing from behavior. Because I think that signatures, you know, until I have a good signature for the next system I want to probe, I'll be very hesitant to put a subject and record their brain activity. But if I could show that my 4 hertz rhythm, that I managed to kind of validate in this very well understood visual domain, helps me understand how, for example, I'm able to do cross-modal experiments where I'm paying attention both to an auditory stimulus and a visual stimulus, and I'm able to show this temporal dynamic. I think that's quite strong, actually, because that is the performance. That is ultimately what, I mean, at least I am interested in. Yeah. The signatures will follow, and I think as we corner these questions through performance, actually, and this is why I think the method, the kind of the innovation that we brought with measuring these things in performance, kind of as we tackle these questions from different directions, we will be in a better position to identify signatures that are not just another brain signature that is thrown at you, you know, across discipline, you know, for you to deal with.
Yeah. I mean, that all sounds totally reasonable. I just think that maybe if that's your view, you should be careful about saying things like attention is this brain rhythm, as if like, you know, there's nothing further to explain about attention once we've found its sort of distinctive brain rhythm signature. Because a few times in the talk, you know, you'd say things like that, and you'd also say like, you know, brain mechanisms use this kind of brain rhythm to do blah, 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 and that's complicated. So, I mean, I agree that this may be really important and part of the whole picture, but just those claims are going to kind of, you know, worry philosophers a bit, that's all.
So we need to finish our session now. We can continue on in the discussion. Please thank Ayelet for a great talk.
Don't worry. Worry too much.